This article looks at the structure and function of the kidney, including its roles in urine production and homeostasis. This is the first in a series of articles examining the renal system. It is read to you using an automated voice, so some of the words and phrases may not be as you would usually expect to hear them. The full version of this article, with citation, references, and illustrations, can be found at nursingtimes.net under the Anatomy and Physiology category. Subscribers to Nursing Times have unlimited access to all our current and archived clinical articles, including a range of online learning units on fundamental aspects of nursing to contribute towards your continuing professional development and revalidation requirements. The full version of this article, with references and illustrations, can be found at nursingtimes.net under the category Anatomy and Physiology, along with our AI search facility, Ask Nursing Times, which allows you to explore the subject further. Abstract. The kidneys are central to the filtration of waste products from blood in the production of urine. Alongside this primary role are several other homeostatic roles, vital to wider physiology. This article examines the external and internal anatomy of the kidneys and discusses the process of urine production and homeostasis of blood volume and pressure through examination of nephron structure. It explores the kidney's contribution to wider homeostasis of red blood cell and platelet regulation and bone density. This is the first in a series of articles about the renal system and is written by James Taylor, lecturer in Anatomy and Physiology at Swansea University. Introduction. Along with the ureters, bladder, and urethra, the kidneys form part of the wider urinary system, responsible primarily for the removal of waste products from blood and their elimination from the body in urine. Found either side of the vertebral column in the peritoneal cavity, the adult kidney filters around 120 nL of blood plasma per minute. The kidneys also carry out several other homeostatic roles, including regulating blood volume and pressure, pH and electrolyte composition, stimulation of erythropoiesis, production of red blood cells, supporting vitamin D biosynthesis. Anatomy of the kidney. External anatomy. An adult kidney typically measures 12 cm long, 6 cm wide, and 3 cm deep. Spatially, however, due to the size and positioning of the liver within the upper right quadrant of the abdominal cavity, the right kidney is typically 2 meters shorter, 8 g lighter, and sits slightly lower than the left. Each kidney is surrounded by 3 main layers. From outermost to innermost, the layers are as follows. Renal fascia, a thin layer of connective tissue that attaches the kidney to surrounding tissue, including that of the abdominal wall, providing structural support. Adipose capsule, perirenal fat that protects the kidney from trauma and helps maintain its positioning within the abdominal cavity. Renal capsule, connective tissue that helps maintain the kidney's structural integrity and shape, protecting its internal tissue. Internal anatomy. The internal structure of the kidney is generally considered to comprise 3 main regions: the renal cortex, renal medulla, and renal pelvis. The outermost region, the cortex, has a granular-like appearance due to the high number of renal corpuscles in this area, each consisting of a glomerulus and Bowman's capsule. The renal corpuscles form the filtration centers of the structural and functional units of the kidney, the nephrons, discussed later on. The cortex also contains a high density of blood vessels, which serve the nephrons. The middle of the kidney, the medulla, contains triangular structures termed renal pyramids, which comprise the loops of Henle and collecting ducts of the nephrons, blood vessels of the vasa recta. Filtrate formed by the nephrons drains through these pyramids, via the renal papillae, into the minor calyces, then the major calyces. When it enters the minor calyces, it is considered urine. The innermost portion of the kidney is associated with the renal pelvis, a structure formed from the merging of the major calyces, and that distally becomes the ureter. Therefore, the renal pelvis channels urine from the major calyces to the bladder via the ureter. The ureter, along with the renal artery, vein, nerves, and lymphatic vessels, pass through the kidney at an indentation termed the hilum. Physiology of the kidney. Nephrons and urine production. Central to the operation of the renal system are the nephrons, of which there are approximately 1 to 1.3 million per adult kidney. Approximately 80–85% of these are termed cortical nephrons because the bulk of their structure is in the renal cortex. The remaining 15–20%, called juxtamedullary nephrons, are on the cortex-medulla boundary. Intrinsically linked to their previously mentioned roles, the kidney's primary function is the filtration of waste products from blood plasma through the production of urine. The major waste products removed in urine are the nitrogenous compounds, urea, uric acid, and creatinine. Metabolic waste products, toxins, excess hormones, water, and electrolytes are also removed. The process of urine formation has 3 main stages. Glomerular filtration. Renal filtrate is produced, containing components of the blood that can be filtered out. Tubular reabsorption. The body recovers 99% of the water and electrolytes from the renal filtrate. Secretion. Waste products such as urea, uric acid, and metabolites are removed through the distal convoluted tubule and collecting duct in urine. Nephrons facilitate this vital process and consist of the following 5 regions. Renal corpuscle, comprising the cup-like tubule of the Bowman's capsule and the dense network of glomerular capillaries, glomerulus. The renal corpuscle is responsible for filtration of blood plasma and the resultant production of renal filtrate. Blood arrives at the glomerulus via the afferent arteriole from the renal artery and leaves via the efferent arteriole. Several structural elements exist within the renal corpuscle that allow for rapid and effective filtration. First, the permeability of the glomerular capillaries to water, small ions and other solutes is approximately 100 times greater than elsewhere in the body, ensuring efficient filtration. There is also increased pressure in the glomerular capillaries due to the efferent arteriole having a smaller diameter than the afferent arteriole, restricting outflow and increasing resistance. Finally, when blood plasma is filtered out of the glomerular capillaries and into the lumen of the Bowman's capsule, it uses a tri-component filtration membrane that comprises the endothelium of the glomerular capillaries, the basement membrane, and a layer of podocytes. These 3 components work together to provide a highly effective means of selective permeability, permitting only those components that need to be filtered. Proximal convoluted tubule. From the Bowman's capsule, the renal filtrate passes into the proximal convoluted tubule. One of its primary roles is permitting the reabsorption of 65–70% of the filtrate's water content, along with several important water-soluble electrolytes, including sodium, chloride, potassium, calcium, and bicarbonate. Approximately 80% of the filtered bicarbonate is reabsorbed by the body from this region, with a further approximately 15% from the loop of Henle, principally the ascending limb, and approximately 5% from the distal convoluted tubule and collecting duct. The reabsorption of bicarbonate ions and secretion of hydrogen ions play a pivotal role in the regulation of the body's acid-base balance, which is crucial to wider metabolic function and therefore normal physiology. Hydrogen ion secretion and bicarbonate ion reabsorption within the nephrons also contribute to the typically acidic nature of urine. The proximal convoluted tubule is the only region of the nephron able to permit the reabsorption of glucose. Any excess beyond this point is removed in urine. Loop of Henle. Following reabsorption from the proximal tubule, the remaining filtrate enters the loop of Henle, a region of the nephron crucial to the kidney's ability to concentrate urine and facilitate further reabsorption of water and electrolytes. This region is, therefore, vital for the regulation of fluid volume and blood pressure, as well as acid-base balance through bicarbonate reabsorption. This region's processes are made possible by its unique structure and differential permeability. The loop of Henle comprises two main regions, the descending limb and ascending limb. The route taken by the loop of Henle, combined with the descending and ascending limbs' differing permeability to water, result in an osmotic gradient within the renal medulla. This forms the countercurrent multiplier system, which is crucial to the kidney's ability to concentrate urine. Starting at the renal cortex, the descending limb drops deep into the renal medulla. A hairpin bend connects it to the ascending limb, which returns to the renal cortex. The initial section of the descending limb is structurally similar to the proximal convoluted tubule and is termed the thick descending limb, while the latter portion becomes narrower and is referred to as the thin descending limb. Likewise, following the hairpin bend is the thin ascending limb, followed by the thick ascending limb. Composed of simple squamous epithelium, the descending limb is highly permeable to water. Therefore, here, approximately 20% of the filtrate's water content is reabsorbed as it moves out of the tubule and into the interstitial fluid surrounding the nephrons. This region is also permeable to electrolytes, including sodium and chloride, allowing for further reabsorption of these crucial solutes. Urea also moves into the tubule for removal. Conversely, the ascending limb is impermeable to water. However, sodium, chloride, potassium, and other ions are transported both into and out of the ascending limb through several means. Ion transport occurs here via active transport processes, including the use of specialized transport proteins. The ascending limb is also responsible for the reabsorption of calcium and magnesium by the body. The active solute transport that occurs here, combined with the water-impermeable nature of the region, contribute considerably to the kidney's ability to both retain water and maintain the osmotic gradient within the interstitial fluid surrounding the loop of Henle. Distal convoluted tubule. Beginning where the thick ascending limb returns to the cortex and passes closely by the glomerulus, the distal convoluted tubule has several key roles in electrolyte and water reabsorption and the concentration of renal filtrate. The distal convoluted tubule, therefore, plays a major role in blood pressure regulation. Central to this functionality is the juxtaglomerular (JG) apparatus. This complex structure is the result of the specialized interaction between three main components, where the start of the distal convoluted tubule passes the afferent arteriole. These three structures work closely together to achieve effective blood pressure regulation through both local and wider means. This is discussed below. The three components that form the JG apparatus are the macula densa, JG cells, and extraglomerular mesangial cells. The macula densa comprises the densely packed, tall epithelial cells of the distal convoluted tubule at the point where it passes the afferent arteriole. These specialized cells of the macula densa act as chemoreceptors, which serve to detect fluctuations in the solute concentration. The JG cells are the specialized granular smooth muscle cells that line the walls of the afferent and efferent arterioles. Containing secretory granules, these cells are responsible for release of the aspartic protease renin, an enzyme pivotal to electrolyte and blood pressure regulation through activation of the renin-angiotensin-aldosterone system, ARAAS. The JG cells act as mechanoreceptors, releasing renin in response to a drop in blood pressure at the afferent arteriole. The extraglomerular mesangial cells, LACES cells, are flat cells found near the macula densa in the gap between the distal convoluted tubule and the afferent and efferent arterioles. Relatively little is understood about their specific function, but they have been shown to be involved in the production of renin and erythropoietin, EPO, serving to stimulate the production of red blood cells. Where there is high demand for renin and EPO production, the extraglomerular mesangial cells can undergo metaplastic transformation, giving rise to different mature cell types that can produce each of these. Collecting ducts. The filtrate then passes into the collecting ducts, whose primary role is to fine-tune the filtrate's composition before it is channeled to the renal pelvis via the minor and major calyces. Adjusting the filtrate here allows for regulation of electrolytes, acid-base balance, extracellular volume, overall water balance, and consequently, blood pressure. The collecting ducts use specialized membrane transport processes aided by hormonal control, making them an important region from which the body can reabsorb sodium and chloride. Reabsorption of these solutes creates a concentration gradient, which in turn promotes the reabsorption of water. In the collecting ducts, approximately 9% of the filtrate's water content can be reabsorbed under the control of antidiuretic hormone. This is a crucial aspect of blood pressure regulation, blood volume, and pressure regulation. One of the primary means by which blood pressure is regulated, and in which the kidneys are a crucial facilitator, is the RAS, produced, stored, and secreted by the JG cells of the JG apparatus. Renin, angiotensinogenase, is an enzyme essential to initiating the events involved in this system. As such, renin is typically considered the rate-limiting step in the process. Through a series of hormonal and enzymatic interaction between the kidneys, liver, lungs, and heart, the RAS is crucial to the homeostasis of blood pressure, extracellular volume, and blood sodium level. With its secretion from the JG cells, triggered primarily by hypotension, low blood pressure, and hyponatremia, low blood sodium, it catalyzes the breakdown of its liver-produced protein substrate, angiotensinogen, into the relatively inactive peptide hormone, angiotensin I. Angiotensin-converting enzyme is then produced, principally within the lungs, and catalyses the conversion of angiotensin I to angiotensin II. The following physiological actions are primarily triggered by angiotensin II and have the net effect of increasing blood volume and pressure. Release of the mineralocorticoid hormone aldosterone from the zona glomerulosa region of the adrenal cortex. Aldosterone acts on the distal convoluted tubule and collecting ducts of the nephrons, causing reabsorption of sodium and water from the tubules. Release of antidiuretic hormone, a peptide hormone also known as vasopressin, from the posterior pituitary gland. Antidiuretic hormone also targets the distal convoluted tubule and collecting ducts of the nephrons, increasing their water permeability and causing reabsorption of water from the tubules. Targeting the hypothalamus, inducing thirst to increase water intake. Vasoconstriction, narrowing of the blood vessels, principally in the arterioles. Hypertrophy, increase in cell size within the heart and vascular system, which eventually causes a reduction in elasticity. Atrial natriuretic peptide and brain natriuretic peptide are hormones that maintain blood sodium concentration and blood pressure, in direct contrast with the effects of the RAS. Both natriuretic peptides are produced principally by the cardiomyocytes of the heart's atria. Their secretion is stimulated by increased blood pressure, detected through stretching of the myocardium. The natriuretic peptides work primarily on the nephrons to increase sodium excretion and therefore water, resulting in a decrease in blood volume and pressure. Red blood cell regulation. The rate of erythropoiesis, red blood cell production, varies between individuals, but it is estimated that average production is 200 billion new cells per day. Central to the regulation of red blood cell production in bone marrow is the glycoprotein hormone, EPO. During the development of a fetus, the hepatocytes of the liver are the primary source of this hormone, but, from birth, demand is met by specialized cells of the renal cortex. Surrounding the nephrons of this region, interstitial peritubular fibroblasts are signaled to produce EPO in response to hypoxia, low tissue oxygen content. This state is typically the result of blood loss, anemia, tissue damage, or high altitude. Renin-producing cells can also be signaled to undergo metaplastic transformation to a cell type capable of producing EPO, helping to meet increased demand in these situations. Once released into the circulation, EPO binds to EPO receptors on erythroid progenitor cells, a form of stem cell. Following binding, these cells differentiate into mature red blood cells within the bone marrow. Binding also promotes cell survival and proliferation. Alongside the erythroid progenitor cells, EPO receptors are also expressed by the cells of other tissues, including those of the heart and the nervous and muscular systems. Research is yet to discover the full extent of the effects on the non-erythroid cells, but studies have shown EPO to promote maintenance and repair, angiogenesis, new blood vessel formation, and stem cell maturation. Platelet regulation, essential to effective blood clotting. Platelets, thrombocytes, are produced in the bone marrow by the process of thrombopoiesis from precursor cells called megakaryocytes. The principal regulator of this process is thrombopoietin, a glycoprotein hormone produced primarily by the parenchymal cells of the liver, but supported by production from cells of the kidney's proximal convoluted tubules and stromal cells of the bone marrow. Via the circulation, thrombopoietin binds to thrombopoietin receptors on the surface of several cell types, including hematopoietic stem cells and megakaryocytes. Following binding, a series of biochemical signaling cascades are set in motion within the cell, resulting in changes to cell proliferation, survival, and differentiation into platelets. Activation of vitamin D and maintenance of bone density. Another key role played by the kidneys, which is often overlooked alongside urine production and erythropoiesis regulation, is vitamin D activation. Vitamin D3, cholecalciferol, is produced by the action of sunlight on the skin, as well as through dietary intake. It is of relatively low biological activity and typically considered a pro-hormone of the more active form of vitamin D, calcitriol. Conversion to calcitriol occurs over 2 stages. The liver first converts vitamin D to calcidiol, then the proximal convoluted tubule of the kidney converts it to calcitriol. Transported via the blood, calcitriol brings about a series of carefully controlled events, with the net goal of contributing to bone density maintenance and overall calcium homeostasis. Some of calcitriol's key effects are acting on the intestines, the duodenum and jejunum in particular, to promote reabsorption of calcium and allow the blood calcium level to rise. Suppressing parathyroid hormone production by the parathyroid gland to allow the blood calcium level to rise. Suppressing calcium reabsorption within the nephrons to maintain the calcium content of the extracellular fluid. Conclusion. The kidneys filter waste products from blood in the production of urine and play a key role in homeostasis. This article explored the structure and function of the kidneys and discussed the role of this organ in human physiology. The nephrons are central to the function of the kidneys and are crucial to the production of urine, electrolyte balance, and blood volume. Renin production activates a cascade of chemical reactions that also influence the homeostasis of blood volume and pressure and electrolyte composition. Wider functions of the kidneys include homeostasis of red blood cell and platelet production and activation of vitamin D in the maintenance of bone density. The remaining articles in this series will discuss kidney dysfunction, including, through an in-depth look at the pathology, the implications for nursing, and current best practice. The next article in the series will introduce acute kidney injury, focusing on diagnosis and staging of progression. To access the remaining articles in the series, subscribe to Nursing Times. Subscribers have unrestricted access to all content on the Nursing Times website. Visit nursingtimes.net for more information.